In this video, I want to talk a little bit more about the electron transport chain, specifically what makes up the chain. The chain is made up of 4 multi-protein complexes that you should be familiar with, or at least have some sort of idea of. So the first complex, basically, we talked about how we're reoxidizing NADHs and FADH2s. Well, in complex 1, we're starting off with NADH. It's coming to the electron transport chain, and it's ready to get oxidized. So complex 1 is called NADH coenzyme Q oxidoreductase. That's a crazy name and all, but I'll break it down for you in just a second. So complex 1, what happens? We have electrons going from NADH, they're flowing all the way, they're making their way towards coenzyme Q, which is also called ubiquinone. Now, why is it called NADH co-Q oxidoreductase? Well, NADH is oxidized, coenzyme Q is reduced because the electrons are going from NADH to coenzyme Q. So, at least, well, not directly, by the way. I'll elaborate more on that in just a second. But the electrons are going from NADH to coenzyme Q eventually. If you know that much, then what would you expect has the higher reduction potential, NADH or coenzyme Q? If you said coenzyme Q, you're correct, because the electrons flow from low to high reduction potentials. So at complex 1, this is essentially what happens. Now what we want to do, what's the point of all these different protein complexes, the protein complexes of the electron transport chain? Essentially what we want to do, this is very critically important, I think, to take reduced electron energy and turn it into potential energy in the form of an electrochemical gradient. So when I mentioned this, I mentioned the proton gradient, right? The proton gradient, where the high concentration of protons was in the intermembrane space, and the low H+ concentration was in the matrix. So what these complexes do, or what we want out of them, is we want these redox reactions to somehow cause the proton gradient to be established. So in this first complex, we want to know if it pumps H+ protons from the matrix into the intermembrane space. So essentially, if we have H+ here in the matrix, and we have a bunch out here in the intermembrane space, do things normally want to go up their concentration gradient or down? If you recall, they definitely want to go down their concentration gradient. So if we want to take these H+ and we want to take them from the matrix to the intermembrane space, that is called active transport, right? Because it's moving against this concentration gradient. And if it's moving against this concentration gradient, it's active transport. That means it's going to require some sort of energy. So complex 1, it actually does pump protons. So first of all, the answer is yes to this question. And how does it actually do that? Well, it has this series of redox reactions in here actually have a negative delta G. Specifically, that negative delta G is, for at least for complex number 1, is negative 81 kilojoules per mole. So that's highly exergonic, and that powers the pumping of these protons into the intermembrane space. Complex 2 is called succinate coenzyme Q oxidoreductase. So we would expect succinate to get oxidized and coenzyme Q to get reduced. That's exactly what happens. The electrons flow from succinate to coenzyme Q. If you recall, succinate showed up in the TCA cycle, right? So we took succinate, turned it into fumarate, and that's actually what's happening here. Succinate is being turned into fumarate, and eventually those electrons from succinate go to coenzyme Q. This enzyme here is embedded in the inner mitochondrial membrane, and this enzyme here is actually succinate dehydrogenase. And if you recall, that's also the thing that makes FADH2. So, and if you recall even further, is that, let me actually write that down, that this succinate dehydrogenase makes FADH2. We'd mentioned in earlier videos that FADH2s are only worth 1.5 ATP, whereas NADHs are worth 2.5 ATP. The reason why here is that complex number 2 does not pump protons into the intermembrane space from the mitochondrial matrix. So because of this, this FADH2 essentially it contributes less to the productive efficiency of this process. So this is a less of a contribution to the proton gradient, right? That results in FADH2 being worth less ATP than NADH. And this process here, you'll notice, is actually much less exergonic than this complex number 1. Complex number 2, it is still exergonic, but it only releases 13.5 kilojoules per mole. So this is a key idea that we'll probably talk about in other videos as well. Complex number 3, the enzymes are flowing from coenzyme Q to cytochrome C. It gets called CoQ-H2 cytochrome C oxidoreductase. What's with the crazy name? And why does CoQ have an H2 on it here? The reason why is because at this point it's been reduced, right? This coenzyme Q was reduced by this NADH ultimately, and when it's reduced, it gains hydrogen. So this is the reduced form of coenzyme Q. But coenzyme Q now is going to get oxidized, and cytochrome C is going to be reduced. And so that's what actually happens at this complex. And this complex's free energy change is negative 34.2 kilojoules per mole. And does it pump protons? The answer here is yes. Matter of fact, all the complexes except complex 2 pump protons. Complex 4 also pumps protons. And it's just called cytochrome oxidase. Why does this one have such a simple name? Well, this is the last complex. This is complex number 4. The electrons are going from cytochrome C, from the previous complex, over to oxygen. So basically, oxygen is just oxidizing cytochrome C. So in this last process, we have oxygen being turned into water, and this is actually highly exergonic. It releases 110 kilojoules per mole of reaction. Now, all 3 of these complexes, 1, 3, and 4, they all contribute to this gradient that we're establishing here, which is very important because we're taking the reduced electron energy of NADH and even FADH2 and turning that into this electrochemical gradient. Now, once we build a high concentration of H pluses around here in this intermembrane space, those H pluses want to get back into the matrix, but they can't unless they travel through a particular ion channel that's called ATP synthase, which we'll talk about later. But you should recall that things flowing down their concentration gradient is a spontaneous process. So essentially, once we use a spontaneous series of redox reactions to power the production of this electrochemical gradient. Now, once these H pluses begin to flow back down their gradient, that's going to release energy, and we're going to use and harness that energy to produce ATP. Specifically, ATP synthase is the enzyme that does that, and we'll talk more about that later. So what about the chain itself? I keep getting all these sort of, simple ideas as far as descriptions of the electron transport chain, but what does it actually look like? Well, this is it. But what's going on here? I drew this little key here to kind of keep things as simple as possible. Anything written in this green color here is complex 1, so that would include these things here. Complex 2 is written in this light blue here. Complex 3 is in gray, and then complex 4 is in this lime green here. Any red arrows indicate a reduction reaction, and any blue arrows indicate an oxidation reaction. Anything also written in white is not really part of a complex. It's sort of like what's it called? NADH and NAD plus. It's sort of part of complex 1, but it's not really all a part of complex 1. CoQ here, notice it's written in yellow. That's because it kind of takes things and is involved with more than just one complex. Same thing as with cytochrome C here. It's kind of in between complexes 3 and 4. where and coenzyme Q is between 2, 1, and 3. So it's kind of a mess. So but that's not all too important. Anyway, what's going on here? So NADH needs to get reoxidized. So it comes in here and starts off the chain. So NADH is oxidized, hence this blue arrow here. But when it's oxidized, something else is reduced. What is reduced? FMN. FMN is a protein, right, that will accept the electrons and the protons from NADH. So NADH is giving up its protons and its electrons to FMN. FMN, you'll notice, becomes this FMNH2. So that the fact that it picked up those H's means that it was reduced. So now it's carrying those electrons and those protons. Now it is going to be oxidized back into FMN, and when it is,